This is episode 291 of the Stem Cell Podcast, Patient-Derived Organoids, with Dr. Scott Younger. Hello, everybody. This is Dr. Daylon James. Also with me is Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Scott Younger. As I was joking with Arun, what a perfect name for a regenerative biologist. He's from Children's Mercy Hospital, and he's on the podcast to talk about his research on patient-derived organoids. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, this is your reminder to register for ICCR 2025. Register by April 29 to get the advance rate. And you can also check out our previous episode to hear from the ICCR leadership and find out what to expect at the conference. Learn more at www.iccr2025.org. We're going to start things off with a stem cell paper. The title here is Vagal Pathway Activation Links Chronic Stress to a Decline in Intestinal Stem Cell Function. This is coming from Shanghai, the Tongji University Shanghai, and various departments in and around Shanghai, different universities in China. Last author here is Mubin Lin. Maybe we'll see some of these folks in Hong Kong, who knows? This is a story connecting chronic stress to intestinal stem cell function. And we know, unfortunately, that chronic stress can really adversely affect intestinal health. And perhaps all of us have experienced this at some point to some level. But the specific neural pathways that actually link the brain to intestinal tissue aren't fully understood. I mean, stress is it's a very broad kind of catch-all term, and certainly there's neurologic stress and there's a connection to all different tissues downstream in the body that are impacted by stress. But here we're really specifically talking about the intestine, the gut, and that brain-gut connection that we've alluded to previously on the show. And, this is sort of a tangent. We've talked about this, I remember, in various papers that discuss like the connection between potentially neurodegenerative diseases and intestinal function, like Alzheimer's connections with intestinal function. So this isn't the first time we've talked about this connection here. So these folks showed that chronic stress-induced activation of the central amygdala dorsal motor nucleus of the vagus, which is a very specific pathway, they call it CA-DMV pathway, can accelerate premature aging and actually negatively impact or impair the quote stemness of intestinal stem cells. So there's your connection between aging, between this pathway, the CA-DMV, and the connection to the intestinal stem cells, right? And they did this predominantly in a mouse model to show this. They showed that the pathway can influence intestinal stem cell function independently of the microbiota. I mean, that's another very hot topic in the field, and we've covered those papers too about how the microbiota can impact intestinal stem cell function and so on. This is independent of that. And the hypothalamic pituitary adrenal axis, HPA, in combination with immune response, the sympathetic nervous system, can influence, again, the intestinal stem cell function. So under conditions of chronic stress in these mice, the vagal activation can prompt the cholinergic enteric neurons to actually release a pretty well-known molecule here, acetylcholine, which can engage intestinal stem cells via this M3 muscarinic acetylcholine receptor, or CHRM3. So a lot of intricate, detailed mechanisms here. And then they further fleshed out the mechanisms to figure out that P38 MAP kinase, another very well-studied pathway, can trigger growth arrest and mitochondrial fragmentation in the ISCs, ultimately leading to that aging-like decline. So this is a complex mechanistic study here detailing a pretty intricate cascade going from the brain, going through P38 MAP kinase, acetylcholine, all these mechanisms, ultimately leading to intestinal stem cell negative impact on ISC intestinal stem cells and this quote aging related decline. So this altogether kind of provides some insights into this neural mechanisms that might be able to link stress to intestinal dysfunction. I think there's a ton that has to be fleshed out here mechanistically. I mean, this is, of course, a mouse model. We got to see how this relates to humans, but and maybe there are some therapeutic approaches here. So maybe you could actually target this DMV associated vagal pathway as an approach for reducing stress induced intestinal diseases. But take a chill pill, everybody. Just cut down on your stress a little bit, do some meditation. So therefore, your intestinal stem cells will thank you for that.
Seriously, guys, save your gut, meditate. I have a kind of mirror to this story, and what's so fascinating and why I love science generally, but this two stories, this one and the one I'm about to present, is how the interrelationship of all these, uh, facets of your existence, um, that converge on cellular function, stem cell function, in both cases, as I'm soon to come around to. But specifically here, the balance of, like, the environmental uh, insult and the gut, right? And stem cells in the gut and the brain, which, as you said, we've talked about that relationship on the show before. But this is one of those studies where you see how all the pieces fit together, and it's totally intuitive when you think about it, right? Like, just like gray hair comes with stress, you would imagine you get some accelerated aging in the gut niche too, given the intimate relationship between the brain and the gut. But here, it's really great to see how they unpack it. And particularly, I think the relationship to patients, while, as you said, a lot more needs to be demonstrated to see whether this is relevant clinically. They did show, descriptively, that there was a increased acetylcholine and P38 signaling in the guts of stressed patients. So I feel like it's not a huge leap um, to make that link, and it makes sense. Also, you hear about people with not just with disease like cancer, but also just stress generally is going to affect the gut. I've been there. You've been there. Grant time upsets tummies all around. But the therapeutic element here, like whether it can be targeted, I think that's where we're going to have to a lot more work before we see someone. I don't know. I mean, apart from meditation, I don't know about like short circuiting acetylcholine or P38, whatever it is. It remains to be seen whether or not we're going to be able to get at this, make it druggable.
Yeah, everybody wants to jump to the therapeutic element for everything. Oh, we got to find a drug. We got to find some sort of target that's going to reduce this intestinal stem cell aging in response to chronic stress. What about the preventative side of things, like you're alluding to? Do some meditation. Don't watch as many close Duke basketball games like we have been watching. Take a chill pill, relax, get some sleep, eat something nice. These things will, I'm sure, that's a cheaper way to approach intestinal stem cell aging, but no one wants to do that these days, right, Dalen?
Absolutely. Although, life happens, Arun. You got a young kid, stress creeps up on you. No amount of meditation can save you from those terrible twos. You've been there and you're coming back around. Good luck, partner. Ha, 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 ha. But that's a good point. I would go even further. I know it's a cell stem cell paper, so you got to talk about therapeutic application. But I love stories like this just because of the mechanism and linking these things that we intuitively assumed to be true. So who cares about the therapeutic action? I just like the way they told this story. And I got a story, as I was alluding to earlier, that is a mirror to this a bit, and it's about the liver, right? And we talk about the liver a ton on this show, and I have a history with the liver. So makes sense, but it is a great representation of the regenerative capacity of mammalians organs. It's the only one really, not the only, but it's one of the few organs in higher vertebrates, mammals, that is regenerative, and that regeneration is governed by proliferation of hepatocytes. And what, I was kind of mentioning earlier in terms of the interrelationship, what we don't really think about a lot is that uh, hepatocyte proliferation and liver regeneration is aligned with the body's physiological demands, right? This is a metabolic organ, and it has to deal with a lot of insults. But understanding the interrelationship between like the equilibrium of physiology and environmental insult and regenerative capacity of the liver is important because, if we want to understand how it goes wrong, perhaps in liver disease, or if we want to enhance liver restoration for patients, who are awaiting transplant, for example. So now getting down to the details of how the liver works, it has this complex vascularity that creates a zonation, right? So blood enters the liver through the portal vein and it drains out through the central vein, and that generates these distinct metabolic zones depending on oxygen diffusion enrichment, and specific hepatocyte populations that go with those zones. So, for example, in zone 1, you have these periportal hepatocytes that are, because they're around that portal vein, they specialize in oxidative metabolism. In zone 3, by contrast, which is poorly oxygenated, you have these pericentral hepatocytes that are they express this glutamine synthase, which converts glutamate to glutamine, right? And zone 2 is somewhere in between those two and has like an intermediate metabolic profile between those two types of hepatocytes. So there's a lot of debate in the field. Some reports suggested zone 3 hepatocytes, right? So those poorly oxygenated zone have stem cell properties. Others have suggested that it's these intermediate hepatocytes in zone 2 that are primarily responsible, right? So this is a story from Nabil Judair, who is at the Centro Nacional Investigaciones Oncológicas. I tried and I kind of failed there, which is like the cancer center, National Cancer Center in Madrid, Spain, that reported on this unique regenerative mechanism in the liver using mice as a model. And what they found is that there's this URI1, which is exclusively co-localized with this other gene, glutamine synthase, in pericentral hepatocytes, right? And by targeting either the glutamine synthase or this URI1 by knockdown genetically knocked down in these mouse pericentral hepatocytes, they show that they could increase the glutamate levels, right? So there's no more glutamine synthase to convert the glutamate to glutamine. So the glutamate increases, and that accelerates liver regeneration, right? And they show, of course, the converse. If you overexpress that URI1, you hinder liver restoration. And here's the thing that I thought was so neat about this story is that you can reverse that phenotype, that suppression or hindering of liver restoration just by supplementing glutamate, or again with the sledgehammer by knocking down genetically the glutamine synthase. Then deeper they go here by showing all the connections and interrelations here, showing that the glutamate, that elevated glutamate, it metabolically reprograms macrophages in the bone marrow, which stabilizes HIF1 alpha, which then activates Wnt3. Of course, Wnt's got to be involved, and that promotes YAP dependent hepatocyte proliferation. So a lot of links in the chain here. And so not just elucidating this kind of elaborate mechanism that connects metabolism in the liver to regenerative capacity, but showing here, again, in these acute injury models, they extend it even more to show that if they add glutamate in these mice, they can enhance hepatocyte proliferation and survival. And then again, just like your story, they move into patients, human patients, and show that these factors, URI1 and glutamate synthase, is co-localized in human hepatocytes, and that correlates with Wnt3, which is the same ligand that was shown in the mice, in immune cells in those macrophages of human patients across liver disease stages. So this is a ton. Not surprisingly, a Nature article, but it's like two Nature papers in terms of the mechanism, the model, and the extension to human. And at the end of it, there's this very simple and straightforward idea of using glutamate as a supplement to help patients who are either awaiting transplant or, maybe have some kind of acute toxicity, or recovering from hepatectomy, whatever. It seems like a really, really basic thing to have a dietary supplement here to improve liver regeneration. Maybe a little bit too easy. We'll have to see how it plays out.
Yeah, these are a couple of really complex mechanistic stories that we've started off the show here with, and we so many pathways to dissect here. You got HIF1 alpha, you got one of my favorite pathways in YAP, you got the whole glutamate axis, and of course we have Wnt, which unites everything and is in every mechanism, it seems like. But I totally agree with you. It's that very last component that I think simplifies the study in a way, and at least when it comes to therapeutic element. It's that glutamate supplementation that could really be transformative for folks who are awaiting transplantation, recovering for hepatectomy, like you're saying. I don't know if it's ultimately never that simple. I mean, the dream is, yeah, you give these folks a pill or supplement, some sort of supplementation, and they'll feel so much better. But glutamate isn't everything. It's pretty much one of the most ubiquitous amino acids out there when you talk about ubiquitous mechanisms like Wnt. So, I think a part of it is also how well can you target this glutamate mechanism. I don't know.
Yes, I mean, I think it's a great point that you appended there. Like MSG, anybody? I mean, don't go out there and get too crazy on the Chinese food, but it might hurt your liver more than it helps it. But I think the important point you're making there is that, yeah, the liver is there to sense the diet and to metabolize, the things that go into our body. So the idea that something so simple as spiking glutamate, while it may work, and I'm not saying it works in mice clearly, and I'm not saying it won't work in human patients, but I think we're going to need a more refined approach to ultimately see the therapeutic payoff. But yeah, I agree with you. It's a nice simplifier. It's a nice cord to wrap around all these amazing mechanistic insights and make it digestible, no pun intended, for someone who's like, okay, what's the upshot here? Why do I care? And I care because, I'm going to go have myself some General Tso's and rationalize it as a healthy choice.
You do that and tell me how that turns out for you. But hey, I'm a fan of General Tso's too myself, orange chicken in particular. But hey, moving on to another stem cell paper. This is coming from, I guess, my neck of the woods, my state in California, a little bit further north, coming from SLA Sharadi. That's their lab is at UCSC, home of the very famous, infamous, world famous UCSC genome browser. Also, the UCSC, what are they? The banana slugs. There are some weird mascots here at the UC schools, I gotta say. First author here is Gerald Lodewick. Apologies for the mispronunciation there. This is sort of related to genomics. I mean, sort of related to the genome browser. I'm sure they use the genome browser here. This is talking about CRISPR activation, another very powerful and useful tool in the toolkit of modern stem cell biology, built on the CRISPR system, of course, but also talking about how they're intersecting CRISPR-A with embryo models, another very hot topic here in modern stem cell biology. Title of the paper here is Self-Organization of Mouse Embryonic Stem Cells into Reproducible Pregastrulation Embryo Models via CRISPR-A programming. I mean, we've talked about these pre-implantation, these very early stage embryo models, a mouse embryo model, but they're trying to make this happen, facilitate this using CRISPR-A, CRISPR activation. And of course, we've talked about this with all the leaders in the world, it seems like, on the show, the power of these early stage embryo models. We've had Nicolas Rivron, we've had Jun Wu, we've had various folks in the field talking about these models. And we know that ESCs can self organize into these structures with spatial, temporal, molecular similarities to natural embryos. No one likes to use the phrase synthetic embryos, so we'll just use embryo models for now, right? And during development, the embryonic and the extraembryonic tissues can differentiate through the activation of a bunch of different endogenous regulatory elements. And of course, the extraembryonic tissue is always perhaps the trickier part of it. How do you get that to develop in combination with the embryonic tissues? There's a lot of cell-cell interactions that have to be figured out here. And here, they're dissecting those regulatory elements, those gene regulatory elements, and their roles in actually facilitating the self-organization of ESCs into these embryo models. Okay, and again, this is UCSC, so they love their genomics here, right? So they use CRISPR-A, CRISPR activation of two specific regulatory elements near a gene that I know very well, GATA6, and also CDX2, to generate embryonic patterns that resemble pre-gastrulation mouse embryos. And then they did the analysis with the live single cell imaging to show that actually self-patterning can happen in a orchestrated movement, is what they called it, driven by cell intrinsic fate induction. So CRISPR-A is inducing cell fate here. And then they did some additional analyses in some 3D models using these CRISPR-A programmed embryo models, or CPEMs, they call it, that actually exhibit morphological and transcriptomic similarity to pre-gastrulation mouse embryos. So you have to do the comparison to the real deal, right? These CRISPR-A programmed embryo models, or CPEMs that they call it, are very powerful because they can be perturbed really easily. You can turn things off, you can switch things off, in particular with those specific regulatory elements that are adjacent to GATA6. So you can do a really mix and match activation of different genetic regulatory elements. But of course, they focused on CDX2 here. And when they induced CDX2 activation in combination with another gene, ELF5, it was able to enhance trophoblast differentiation and lineage-specific activation of certain extracellular matrix proteins that are really important for this whole process to happen, like laminin, matrix metalloproteinases, stuff that we've talked about before. And so that's a whole separate element of the story. It's not just the intrinsic cell fate side of things, it's how the extracellular matrix is actually really important to make these embryo models happen and making them develop, right? So they're figuring out the role in the basement membrane remodeling in embryo model morphology and making these things happen. So there's a lot to dissect here. I mean, I think big picture, it's a reflection on the power of the tool. It's a reflection on the power of the CRISPR-A, to show that intrinsic editing, epigenome editing, can self-organize these ESCs into programmable pre-gastrulation embryo models with really robust lineage-specific perturbation. CRISPR-A enables all that. It allows them to dissect the mechanisms of how these embryo models actually form really nicely. And like I said, this is right in the wheelhouse of UCSC, UC Santa Cruz. It's not just great beaches, it's not just great surfing, it's great genomics too. So check out what they're working on here.
Yeah, and the cell engineering element too, right? So I when I first saw the title of this and read the abstract, a part of me was like, wow, aren't we kind of moving farther away, further away? Ironically, from a kind of what the embryo model was aiming at, right? It was a more physiological proxy of whatever, in this case, mouse development. But now, as you describe it, and as I read deeper into it, my mind has changed because, I mean, I guess, the undercurrent of the whole embryo model field, that I don't know that we don't talk about it, but we all know, is that none of these embryos are capable of becoming actual conceptus or fetus, right, in the mouse or human. And not just because they lack all the extra embryonic tissues, but just like the organization of the thing, it's not true to the ground truth that happens reproducibly in utero, right? And I think a big part of that is that it's hard it's a moving target because all of the embryo models that we're working with in the field, collectively, not myself, are subtly different depending on these unknown unknowns. And this approach here is how you can kind of flatten that out, and get obviously it's in the title, reproducible model that then you can use for gleaning some insight. And here, that's what they did here, as you said, in terms of the matrix, and metalloproteinase, whatever they learned about physiological embryo development. And I think all these things are going to feed forward to refine the models more and more to the point where we will actually have a very reliable proxy of mouse and ultimately human development that is observable.
Yeah, I think we're definitely moving in that direction. And, various folks around the world are certainly refining these models, making them more one to one with the real thing, but definitely not one to one quite yet. CRISPR technology is just one example of how you can interrogate what goes wrong potentially in these models, how you can make them right, make them better. And they're still using somewhat old school tech in CRISPR, if you can believe it, and which has only been around 10 years. But there's a bunch of new CRISPR Cas ortholog systems like Cas12a that allow you to actually further multiplex some of these edits. So there's definitely ways you can make these even better. The other thing is the timing of all this. They only examined the development of these CPEMs for 3 days, which is like nothing, right? So you could extend that way down towards the course of mouse development. So I think a lot of questions are waiting to be answered here, and not nothing to say about the technologies that can be improved on, but it's a nice paper that represents the integration or the intersection of multiple hot technologies in the field of stem cell biology.
Yeah, that's for sure. Super tech. Well, I got some that's it's like Back to the Future, I guess. In terms of, one of the original targets that we talked a lot about in therapeutic translation was the eye, right? Immune privilege, and a lot of progress was made with RPE in the early days. And not that it stalled, but of course, like everything, it takes time. Um, but this is a story that was a shocker to me because I thought we kind of worn out all that we knew about stem cells in the eye. And it turns out not really, not close. And there's a lot more to learn, and we got tools now to address it, not just experimentally, but therapeutically. So yeah, talking about it from a disease standpoint, retinitis pigmentosa, macular degeneration, leading causes of blindness globally, as well as other less prevalent diseases. But it's a problem, and there's no real treatment or cure, of course. And the basis of the pathologies is irreversible degeneration of either the neural retina, or the retinal pigmented epithelium. We hear a lot about RPE, but not as much about neural retina, or both. Sometimes both of those are going bad. Um, the treatments now, it's just like, let's try and stop or slow the degeneration of the neural retinal cells and protect what's left. But there's not really a regenerative therapy that is effective for these neural retina cells, these NR cells. Of course, stem cells would be great, but there's not a lot of donor cells for neural retina that are regenerative or proliferative potential when you talk about from cadaver, um, or other sources, even stem cells in culture. Um, so you know retinal stem cells are known to exist. They generate neural stem cells, but their existence in the human retina, it's debatable, right? We know that these things exist in zebrafish, of course, and other amphibians, not other amphibians, but amphibians in addition to zebrafish, that have crazy regenerative capacity, not just all over the place, right? But in the retina specifically, where they have these specialized circular stem cell zone, that is at the interface of the neural retina and the retinal pigmented epithelium. It's called the ciliary marginal zone, which may be familiar to some of you. And in that zone, there are both these retinal stem cells in fish and amphibians, as well as these retinal pigmented epithelial stem cells, right? And it's known that these retinal stem cells, they self-renew, and they continuously differentiate to form new neural retinal cells. But they're present in higher vertebrates. It's debated, still uncertain. But nowadays, we have the tools, right, to really interrogate all the cell types with tremendously subtle and fine resolution with single cell, right? Not just single cell, but spatial transcriptomics. And also, we have human retinal organoids, which allows us to come, from the ground up, try and generate these human organ enlages, in this case, the retina, where we can try and isolate and identify the unique stem cell types. So that's what Jiaqu, the groups of Jiaqu and Zhang Shu did, who were at Wenzhou Medical University in China. This is a story in Science Translational Medicine. And in those labs, they leverage these two technologies. First, they use single cell multiomics and spatial with human fetal retinas. They had a ton of human fetal retinas from around 16 to 28 weeks, more than 10 of these fetal retinas that they use for a lot of purposes. And they use the multiomics and the spatial to show that there are indeed human neural retinal stem-like cells. They call them stem-like. You can't call it a stem cell in any of these papers. Stem-like cell. Although if it's in the fetal retina, I would say we can take the like off of it. This is bona fide. They show that they reside in the ciliary marginal zone, similar to the amphibians and fish. And they self-renew and differentiate. And this was painstaking. I mean, they did a lot of descriptive stuff. They did all the pseudotime and to try and track, but they also took these cells out and showed they had clonal potential. They did, seek then of day 0 versus 3 weeks of differentiation in vitro. That's all fetal tissue, show that there was a progression. And then they leveraged the other tool, which is the human retinal organoids, which these labs had a part in generating the primary stories demonstrating the presence of these human retinal organoids from pluripotent stem cells. And they showed, again, using single cell, that they have these similar NRSCs, these neural retina stem-like cells, with a similar profile, transcriptionally similar developmental trajectory. And then mechanistically, of course, they show that there's these critical transcription factors, for example, MECOM, that governs the differentiation and retinogenesis from these cells. And then, of course, they transplant these cells, because it's a Science Translational Medicine paper, into this mouse model of retinitis pigmentosa, and show that they differentiated and integrated into the retina, alleviated the retinal degeneration, and improved visual function. So curing these blind mice, at least 3 of them, and a story that I think resonates pretty tremendously in the field of all these people who've been focused on the eye, providing a new avenue for retinal regeneration that could have a massive impact. Everyone, what do you think?
I mean, first of all, I love science translational medicine, and not just because I publish there, but this is a story that kind of exemplifies the type of work that's in STM. I mean, this is awesome. So you go all the way from an observation in human fetal retinas uncovered via single cell RNA-seq, right? And then you go into the organoids, the human organoids, and find that that same population is present in the organoids. And then you figure out some repair mechanisms in the organoids. And then you take these cells and put them into a repair model in a mouse. That's amazing. From very basic deep dive single cell RNA-seq, figuring out the cell population, and then ultimately transplanting those cells into a repair model in the eye for retinitis pigmentosa, or retinal degeneration. That's incredible. And I think the next step here is obvious. Taking these into other models, they actually alluded to this, that the mouse may not be the best animal model to study this sort of retinal degeneration. They actually refer to a rat model, which may be superior here. You have other larger animal studies that you could be doing. But regardless, I think just this is a study that really exemplifies the quality of science that's in science translational medicine. We do cover these stories every once in a while, but one of my favorite journals, phenomenal paper here.
Agree. And if you get a stem cell story, particularly like a pluripotent stem cell story in science translational medicine, it's going to be a great read because it's like this. Um, going to be one where you're like, wait, really? Okay, fire it up. I mean, let's go. When do the clinical trials start? Or at least, as you're saying, when do we go into larger animal models that can really test the capacity of these cells? Because it feels like a watershed for the field. So very exciting, and I think emblematic of the exciting times we're living in, where we're starting to see real light at the end of the tunnel, in this case, pun intended, for some of these cell-based therapies. And our guest today has similarly leveraged human pluripotent stem cell-derived organoids, patient-specific, to glean some insight into rare disease. And we're going to talk a bit about that in just a second. But first, I got a quick message from STEMCELL Technologies. Are you interested in exploring the applications of organoid cultures? Get a free wall chart showing how organoids are used as model systems to study infectious diseases, cancer, congenital disorders, and tissue regeneration. To learn more, visit stemcell.com/organoids-modeling-wall chart. All right, everybody. With us today, we have Dr. Scott Younger, who is Director of Disease Gene Engineering within the Genomic Medicine Center at Children's Mercy Hospital. Since joining Children's Mercy, Dr. Younger's lab has pioneered methods for the rapid, robust, and scalable production of patient-derived cellular models. In the lab, they're currently focused on the development of platform-based approaches for functional precision medicine, and with an emphasis on reduction to practice and clinical implementation. Dr. Younger, thank you so much for joining us today on the show.
Thank you so much for having me. I'm excited to talk to you about some of our work.
Yes, we'll have to jump into that reduction to practice in just a sec to clarify. But just again, by way of introduction, we love showcasing the technologies and research that are moving the field. And your work has so many elements that are at the leading edge, from the large-scale modeling of rare disease biology with iPSCs to precision medicine, as we alluded to in the jump there. So why don't you start us off by giving our listeners an overview of the overarching goals and means to those ends?
Yeah, so I think I need to tell you a little bit of a historical perspective about the lab, which opened about 6 years ago when I came to Children's Mercy. And around that time, we were launching a rare disease initiative here at the hospital, a program we call Genomic Answers for Kids. And in that program, we're aiming to sequence the genomes of roughly 30,000 children impacted by rare genetic conditions. This is largely undiagnosed rare disease. So when my lab came in, we were really focused on building out one of the research components of the program. And so really using genome sequencing to explore genetic variation in our patient population. And one of the things that came about in our work, we kind of had started with the initial idea of we'll just buy some off the shelf iPSC lines and engineer in variation and study it. And it turns out that's really tough to do. CRISPR technology is super cool, but it's not kind of super efficient yet. So we found that it was actually really hard to engineer these variants. And so we turned our sights on producing iPSCs from our actual patients. So that was, rather than trying to engineer variation, let's just harness the variation that we have. So when patients enroll in our in our program, they provide a blood draw for genome sequencing, and we cryopreserve some PBMCs or blood cells in that process. And we envisioned a path where we could take these cells and reprogram them into patient-derived stem cells. So this is a technology that's been around for almost 20 years now. So surely we should just be able to contract that out. But when we started making phone calls, we found that process actually cost about $10,000 per line, or between $5,000 to $10,000. Some of the turnaround times were up to a year before you get, a cell line back, right? And that's per cell line. And so we're sitting with kind of a biobank of tens of thousands of blood samples. And so it's not really, scalable to do one at a time at $10,000 a pop. So we really, the lead author on a paper we just published, which we can talk about in a few minutes, through just a Herculean effort, just put in the hard work to go through the literature, find out how people were doing this reprogramming, what they were doing, what was working, what wasn't, and really distilled this down to a highly optimized protocol where we were able to take residual peripheral blood mononuclear cells and reprogram them into iPSCs in about 2 to 3 weeks, in a process that costs, really about $200 in reagents. So really a game changer for our ability to generate these things. And we've never looked back, right? So we can generate these things in a scalable way. So, a typical research lab might engineer or generate, a handful or dozen of these lines over the course of a lab. We've generated, over 400 of these lines just in the past year, and for various reasons. So that's what's kind of what we're pushing things forward is getting this practice up and working in a scalable and cost-effective manner. So that's a big part of it, and then really using these lines for various purposes, diagnostically or precision and personalized medicine, which is where we recently published a paper kind of on the personalized medicine front. And in that particular study, we focused on N-of-1 therapeutics, or personalized antisense oligonucleotides. So these are also, this is a technology that's been around for 50 years, right, in the laboratory, but it's kind of having a renaissance due to its application in the clinic, and really some pioneering work from Tim Yu's lab at Boston Children's, where they were able to generate a personalized antisense oligonucleotide for a rare disease patient and actually administer this therapeutic and show that it worked, right? Which is really the first time from soup to nuts this whole process had been done before. So what we did was leverage our pipeline, where now we can just turn to our patient population. We had patients with genetic variation or disease-causing variation that would be amenable to such therapies. So we were able to show that we could use blood lines or blood cells from those patients to engineer iPSCs, and then grow those into disease-relevant cell types. So that's the power of the iPSCs, is we can differentiate these into virtually any cell type in the human body. Our lab has also really gone down the path of using organoids, which are three-dimensional cell models, and they're really cool because they recapitulate, organ development, organ function, and what we see is we can actually recapitulate clinical phenotypes in a dish using these organoids. So for the paper that we just released, or was just published in Nature, what we had done was take patients with Duchenne muscular dystrophy, right? And we can generate cardiac organoids from those patients. So that's kind of maybe not totally obvious. A lot of us, when we think of muscular dystrophies, we think of skeletal muscle, but really our heart is kind of the major muscle of our body that keeps us churning. And so when you generate a cardiac organoid from a healthy individual, it just contracts in a dish. It's really mesmerizing to watch these things. But when you generate it from a muscular dystrophy patient, they just kind of sit there. And they'll occasionally move, but you can see it's a qualitative observation, kind of a binary works, doesn't work. And what we found is we could treat these with personalized antisense oligonucleotides targeting disease-causing variants, and reverse that or restore the contraction phenotype within a dish. So really showing that we could go from a blood draw to designing a personalized antisense oligonucleotide to showing functional restoration in a dish, and kind of managing to get that whole preclinical workflow, rather than down to years, down to more on the scale of about 6 weeks, and again in a scalable manner. And so we mentioned reduction to practice, which is kind of more in the patent area that we think about terms like that, but really what that means is taking a workflow, a pipeline, or a platform like we've developed, and not saying, hey, look what we did once, maybe one day we could do this over and over again and scale it. We've actually built the whole platform in a way that's scalable from the outset. So by reduction to practice, if we had 100 patients come through with identified genetic variants, we could get through this process, in a couple of months for all those patients. So we really want this to work kind of out of the gates and not be something that we're building down the line. So that was long-winded, but I'm happy to chat more about anything, any component of that.
Yeah, I mean, I think one thing that's unifying all the work that you're doing recently is this pursuit of efficiency. And efficiency is not always the name of the game when it comes to iPS production, when it comes to organoid differentiation. It seems like that's one of the major things you're trying to tackle, right? Making iPS production or reprogramming more efficient, cost effective. And same thing with the cardiac differentiation side of it. Like, I'm a iPS cardiomyocyte biologist by training as well. I also, do cardiac organoid differentiation. And one of the advantages inherently of that particular cell type is that the phenotyping is so fast, right? You can see within a week if these things are actually contracting, if your differentiation has worked. And so all those things combined together, I think, makes a really efficient workflow and makes a lot of sense for what you're trying to pursue in terms of, personalized precision medicine and modeling. So, you talked about a whole slew of different technologies there, iPSCs, organoids, all, great things that we cover on the show all the time here. And, I wanted to hone in a little bit on the patient specific organoids in particular, right? We talk a lot about this a lot on the show, whether they're iPS derived or primary. These patient specific organoids, like you were alluding to, have this amazing, incredible potential in personalized medicine for drug discovery, disease modeling. And yeah, we'll dive into the Nature paper a little bit in a little bit. But could you talk just a little bit more about that power of the patient specific organoid and the dichotomy between the iPS derived and the primary organoids for the work that you want to do, especially when it comes to drug screening? Just talk about that for a little bit.
Yeah, it's fascinating. Like, it's one of there. I've never been. I love science. I love what I've done, but this is really an exciting arena to be in because every day it's something new. And so, we talked about that cardiac work that was published, but to get to your question, really, what this has really been telling is more in the work we've done in neuronal organoids. And to kind of give some background there, we're working almost exclusively in brain organoids now. And kind of as you sort of alluded to, one of the reasons we got into this space is because at the outset, we wanted to study neuronal development. But if you do a 2D differentiation, you kind of have to make a lot of decisions, like what are we going to make? And when we're sitting, with a pediatric data repository full of patients where we don't know exactly what's going on, right? You're kind of almost felt like we were setting ourselves up for failure to try and make that decision. Whereas the organoids give this complex mixture of stuff. And we felt like we could see phenotypes much. We're more likely to catch phenotypes. And this has really been fascinating. So we have one of our grants that we're focused on, and again, in association with our rare disease program, is focused on development in patients with structural brain abnormalities. And what we find, if you generate a brain organoid from an unaffected individual, you get this nice, tight, three-dimensional ball, brain organoid. And you can do stuff with that. We do a lot of calcium flux assays where you can actually put it on a dye and you can watch these little blips light up. And that's, calcium's part of our neuronal firing action potential. So we use it as a proxy for neuronal activity. We're actually migrating over to these microelectrode array machines that actually directly read out the electrical activity, but you can profile this and quantify it. And so with the brain organoids, what we've seen is when you generate brain organoid from a patient with structural brain abnormalities, they actually form different shapes, even at the level of a three-dimensional ball in a dish. The sizes are different. Some of them will bleb in ways that are unique to that patient. And so you can actually see with your eyes that things are they are developing differently than in a healthy, and I hate to use the word healthy, but we'll say unaffected by disease individual. And so where we're spending more of our time these days is focused again on precision medicine. And so again, back to those brain organoids, if you generate a brain organoid, and this is also where we're kind of going a little bit against the grain, some labs will grow these organoids for months on end to really try and get this developmental, understanding of what's going on over time. We're a little more focused on how quickly can we get a functional readout off of these organoids. So in about 4 weeks of growing a brain organoid from a patient, you'll start to see these little blips if you're using, a calcium indicator dye, and they're sporadic and somewhat rare. If you generate a brain organoid from a patient that's impacted by seizures, these things light up all over the place. It's like a light show. We've done this from now about 4 dozen different patients, and 100% of the time, you can see this neuronal hyperactivity that's specific to patients with seizures. And that's regardless of really, the mechanism of disease or what's going on. It's almost just like a universal lack of inhibition on neuronal firing. And so we were interested so seizures are interesting because we can quantify those. They're highly prevalent in the rare disease community. I mean, in our rare disease program, which spans, lots of undiagnosed rare disease, about 80% of our patients have seizures, right? So this covers everybody. So you can try and treat seizures symptomatically with antiepileptics, but these drugs have a high degree of patient-specific responses. So there's like 30 or more of these things that are FDA approved. Only one or a small subset works for any given patient, and really you have no idea which one it's going to be. So you'll hear about rare disease patients or parents or families talk about this therapeutic odyssey, where you kind of try a drug out for a few months, see what happens, adjust, try a few months. We have patients in our program that have been in this odyssey for over 10 years to try and find the right anti-epileptic. So we were curious, what happens if we generate these organoids and treat them with anti-epileptics in a dish? And so kind of that work, what we saw was, if we just kind of went in a pilot experiment and treated a bunch of different lines with a bunch of different anti-epileptics, we saw right away a high degree of patient-specific responses. And interestingly, you could take 2 drugs that supposedly work through the exact same, inhibit the same enzyme or same mechanism of action, and they behave very differently across patients. And so we were seeing that. And so we've gone on to do a retrospective study, again, through our Genomic Answers for Kids program, and saying, okay, here we have patients that have been treated with these different drugs, and here were their clinical outcomes as recorded in their electronic medical records. And what we found is we could really robustly recapitulate those clinical responses in the dish, right? So we can see this is what happened in the patient. We see the same things in a dish. And we've actually moved that forward on a case study for a patient where the clinician believed that the seizures were being caused by a variant in a specific calcium channel. So we tested out several inhibitors or calcium channel blockers, I'm sorry. And we found that of the whole panel we tested, only one of them worked. They ended up administering that to the patient, and it resolved outward, facing seizures for the patient. So we've kind of gone through a pilot study, through a retrospective, through a case study, and now we're kind of interested in scaling that up and saying, hey, can we screen through drugs in a dish and provide treatment plans for patients? And that's kind of, we're not doing that just yet, but we're lining up, getting things in motion to be able to do that.
Yeah, I mean, I love what I love, I think, the most about this work is how it disrupts this traditional pharma paradigm, right? The whole idea that there's a lot of research that goes into each individual. You got to recoup the cost, so you can only invest in those diseases that have a sizable market share, right? And I think that's been an impediment and tragic, really, because some of these rare diseases maybe are tractable, maybe can be addressed, but there's just not enough money to be made, the cynical part of me says. So it seems like that's not the case anymore for a whole segment of that, unmet need now. That there's these personalized approaches of very varying types. And you just described, I think, in detail one that kind of fits within or can be kind of conjoined with the traditional paradigm of pharma, where you have the screening approach, right? And you have a panel and you make it patient specific in order to see which of the existing drugs, right, will work or will work best. But you alluded earlier to the nature story, which is kind of a different approach in that the drug is personalized, right? You have personalized ASO. So you did talk a bit about the science there, but I want to drill down a little bit more on what does that look like? I mean, you talk about the reduction to practice, I think is what we
Yeah.
call it. Like in terms of how would that be deployed for each individual case? And you can circle back to the science in order to fill in there. But how would that fit within I know you say one day soon, and that's where we were, and you're in this intermediate now where we're trying to actually make it practical, but on an individual scale. What does that look like?
Yeah, so kind of this back and forth between precision versus personalized medicine. So in the paper that we're referring to, that was all based on ASOs and personalized medicine. And in doing that study, we kind of blew right past the precision medicine, which is actually much easier to implement because, like you stated, we're working with FDA-approved drugs. We're actually just providing a clinician with information, right, to make a decision. Whereas the personalized, a drug that's a drug that doesn't exist yet. So how do you go from, hey, here's a challenge, can we take on this challenge, and how does it end up in a patient? And it turns out that's really, really hard. And if you look at kind of the proof of principle, which Tim used work, you have a patient that's really in need of this therapy, a highly engaged family, a really robust health system, and a great investigator. All these things kind of coming together to make this happen. Now, kind of scaling that and democratizing access is really hard. And so there's two major hurdles to the personalized therapeutics approach, at least with ASOs. So the first is the cost, right? So you're talking about depending on who you ask, somewhere between $5 to $10 million to get a lifetime, treatment with a drug or a personalized ASO, and that's kind of mashing everything together because different drugs require different administration, protocols and issues like that. But you have to pay to make it, and so you'll see, some groups are kind of going towards that middle ground of not personalized ASOs, but like ASOs that can kind of treat a larger subpopulation of rare disease. So maybe you can get some, economy of scale that makes that worthwhile. But the money thing, I think, will resolve itself over time. The real challenge, I think, right now is the regulatory burden on these things, right? For the proof of principle with Millicent and Tim Yu's work, you have a patient that unfortunately was terminal, right? And so you fall into a lot of these compassionate use and all these different categories that make that possible. But let's turn back to the muscular dystrophy ASOs that we generated. Again, that was totally preclinical. That work is at the moment not moving forward towards the clinic. And again, backing away, you have patients that have a degenerative disease. These are children that will ultimately succumb to the disease. But as of now, they're on a spectrum, right? And maybe in the middle or the middle of that spectrum in terms of their disease progression. And so they're not at the moment terminal. So they don't fall under that. And then you have kind of this exploratory medicine, right? That's never been used. So is that appropriate? And so all of this kind of gets been together. And so I think figuring out the regulatory approach and kind of breaking it off of kind of the standard workflow of how we work through the process of getting approval to pursue these kind of therapeutics is something that needs to be addressed. And there are groups out there that I think are addressing each of these components. And what's really important in breaking down the rare disease, aspect of this, which I think is really interesting and exciting to be in. Like I'm in Kansas City. There's no shortage of rare disease population here. There's no shortage in any city, right? So this is not a centralized problem. Everybody needs to be addressing this in every city, every research organization or university has their own patient population to work with. So we're working with ours, and that's kind of filling out our workflow. Now, we're not addressing the regulatory component, but as I mentioned, there are other groups that are focused exclusively on working through how do you get through this regulatory system in an effective and efficient, timely manner. And then you have other entities, things like NLORM. So if people aren't familiar, NLORM is a kind of a spinoff of Ionis Therapeutics, and their kind of model is offering personalized ASO treatments for life for patients that are admitted to their program. So really showing, hey, we can do the production of the drug, and we can, get these things out there and show that they work. So that's kind of addressing that component of it, less the research. So we can enroll, we do enroll or try to enroll patients and enroll them. So we feed our patients there if they can be treated. So I think there's different places all over the country, all over the world, that are trying to chip off their pieces. And our piece that we see, our challenge that we addressed was really the preclinical aspect of how can we do this in a way where if we had 100 patients line up today, we could have 100 preclinical leads in 2 months, that once everybody works out all those downstream things, it all just clicks into place.
Yeah, I mean, I do want to hone in a little bit more on the financial side of it, because that is important to discuss here. I mean, it's something we've talked about a lot on the show. I mean, a major issue with a lot of these high power translational technologies, like what we're talking about here, patient specific organoids, patient specific high throughput phenotypic screening, is that these things are in general very expensive. I mean, we've discussed a lot about on the show about some of the pros and cons of the patient specific versus what you alluded to, the, for a second, that off the shelf allogeneic approach for developing therapies, right? I mean, like what we're talking about, the patient specific is perhaps superior scientifically, it's more targeted, individualized, but financially it can be challenging. And every single IPS therapy company is running into this problem, right? Like pros and cons, balancing out the finances of the off the shelf versus the true personalized patient specific IPS therapies. So could you talk more about that balance, about the science, the efficacy versus the economics of some of these therapeutic approaches? I mean, ultimately, we want to bring these cutting edge approaches to everybody who needs them, whether it's on an academic setting or industrial setting, whatever. But how do we do this in a cost effective way? Could you talk a bit more about that?
Yeah, I think a lot. So I think there is one major distinction. We're not doing cell-based therapies, right? So we're not engineering novel therapies that are going back into patients. We're using these as screening systems. So there's a little bit of a difference there. But again, going back into the cost of doing these things, the reason that we got into doing patient-specific modeling is because it was just too expensive and time-consuming to do these off the shelf and engineering these novel cell lines, right? To the end of the day, it's just too inefficient. You need too many specialized people with expert, that's more expensive than just taking some blood, cells, putting them in a tube. And that's reducing. Now, what we're doing, that's really simplifying it, but really, we've gotten this process working in a way where it's pretty basic. I've had multiple people come through the lab, ranging from undergraduate students through PhD scientists that are able to work through this process. So it's that efficient. So I don't think the cost. I don't think that argument is real in terms of those two different approaches, and that's a big thing we've hit on. Now, what you're doing with these models is, I'm sure that can change how much you're going to be paying. But then one of the things that's actually, I think in the research world, we don't think about too much, or maybe I haven't before, but working in a hospital is really enlightening. We're focusing on seizure disorders, and if you look at even rare disease, if you look at how much time these patients are spending in the hospital, the impact on the family, the costs for the family, the impact on the healthcare system, right? So engineering these patient-specific approaches that resolve issues quicker is actually a huge money saver, and it's financially more effective for a healthcare system. We don't see that if you're sitting kind of, and I don't see that day to day sitting in the lab, right? But when you kind of back out and get more of a patient-centric perspective, you realize that this is actually, it helps everybody all the way around.
Yeah, there's such a broad spectrum, and I think it underscores the amazing impact that cells in general have had peripherally on therapy and research. We're talking about these subtle distinctions that are really important between generating a cell line as a model versus generating a cell line as a actual therapy, right? For cells versus ASOs and existing drugs. So there's an amazing and rich spectrum of approaches that's really exciting, as you said at the start. It's such an exciting time to be in this field. I think underpinning all of this, though, is the data, right? When we get into these personalized approaches with individuals and their genomes, really, which is governing and epigenomes, which governing the response and the pathology. It's a lot. I think we all agree that having that as a resource is as square one is critically important, and that's a lot of what your work is focused on. The volumes of data and parsing it to get information, right? We're approaching now more than 500,000, or we're past 500,000 human genomes that have been sequenced and that are out there. And with those kinds of numbers, the potential to understand the genomic basis of disease is really profound. But again, how do we parse that data? You focused on that. Tell us a bit about that. I know you have this scientific reports paper, and there at Children, there's a huge range, as you said, of specific genetic disease. So tell us a bit about your efforts to identify and parse genetic disease on an individual level. And also, and speaking of how do we wrap our arms around this as it continues to exponentially grow, is there a role for, these new tech you see, like AlphaFold was a really great example, or other kind of AI related analysis that is going to make it easier or autopilot some element of the work that you've painstakingly done to really identify and pinpoint some of these rare genetic diseases?
So I definitely don't want to like shoot down AI as being a great tool. I don't think it's going to solve all our problems out of the box or anytime soon. So you referenced a scientific reports paper we published a couple of years ago, and this again. So we have this huge pediatric data repository full of rare disease genomes. That data is available through controlled access through like dbGaP. So all that information is being pumped out to the research community. But one of the things we're interested in doing is again, we focus on the scalability. Can we profile genetic variation at scale? So this is stepping back away from patient-derived models, but we can build high throughput synthetic systems to screen through genetic variation. So we've done things called massively parallel reporter assays, so where we can take regulatory or variants that we believe have the potential to impact, regulatory functions of a gene. We can plant them in front of a reporter, toss them into some cells, and see how that genetic variation, kind of the reference allele versus the alternative or disease allele, do those function differently? And that's kind of what that paper was about. On the regulatory aspect, which falls more into my like personal scientific interest, I think that's fascinating. In terms of therapeutic relevance, that's actually a really challenging space to focus on regulatory variants. Now, what we've done more recently and falls more in line with what we're doing is we've modified the massively parallel reporter assay approach to a mini gene splicing assay approach. So this is super basic, where we have a GFP reporter gene and we just broke it into 2 exons. Right. And we have a small intron that separates those. And then we have a cloning site where we can drop in a genetic variant and see what happens. So, for example, the allele that we targeted in the Nature paper, we drop that allele into our system. And what we see is when you have the reference allele, the GFP splice is fine. And then when you put in the single nucleotide variant in that patient that actually alters splicing in the patient and causes DMD, we see that splicing event happen in the reporter gene. We actually see that splicing event happen at single nucleotide resolution. So we can actually pick out where that splicing event occurs, which is super important. Now, we can adapt that. Instead of looking at one sequence, we can look through tens of thousands of sequences simultaneously, and using kind of a multiplex barcode readout, now we can read out splicing outcomes from thousands of variants in parallel. So we can take, big portions of our rare disease patient population and screen through intronic variation. So why would we care about intronic variants? So going back to kind of our basics of genome biology, you've got 3 billion letters, about 2% of those or less encode for protein coding genes, kind of what we focus on. About 35% of that space is introns, the intronic information in between these exons. And we can look at genetic variation in those introns, and if it falls in very specific sites near exons, we can kind of predict what happens. But for most of that space, we have no idea what the consequence of genetic variation is. So we want to screen through that, and that for a couple of reasons. So we want to understand that space better. Can we identify novel variants that seem to be causing splicing aberrations? And conveniently, that type of aberration is amenable to treatment with antisense oligonucleotide therapies, which we also showed that we can do in the lab. And so we've run those assays, right? And so we can find, genetic variants that do cause splicing aberrations that we never would have predicted based on any other metric. And we actually run those kind of, here's this variant in the empirical data from the assay, here's this variant from kind of Splice AI or other AI-based algorithms or models out there. And what you see is just a complete break. Like there's almost nothing on the axis. They don't overlap at all. And that's not to say that one's better than the other, but they identify different things. And so when we validate this, we've actually gone in and we've focused on variants that we see empirically altering splicing in an assay. But now we can go back to the patient cells. We can generate the iPSCs. We can generate organoids. And so for that patient we spent the most time in, that patient has seizures, right? So we can generate brain organoids. We see the seizure phenotype. So we can design ASOs targeting the splicing variation that we identified, the splicing variant that we identified in our assay, and we see a complete resolution of the seizure phenotypes. What's even cooler with using this technology, or maybe not cooler, but also cool, is that when you generate these brain organoids, this particular patient has structural brain abnormalities as well. But if you treat with the ASOs before you do the differentiation, they form just fine. So you can actually prevent disease development in the model. So it's super, super powerful from a diagnostic standpoint. But the same tools we're using to validate that variation as being causal is inherently a preclinical lead. So that's kind of where we are in the process. But coming back to that AI question, AI is super cool, but especially in research, sometimes we have a tendency to train these models on things we know and then get really excited when they find things we know. Whereas a lot of the things especially in the intronic space, the non-coding genome, we just don't know. So we don't have the proper information for these models to learn how to understand. But we will one day maybe, but based on how much effort has been put in thus far, I don't think it's going to be tomorrow that we have all of those answers. That's not that work should still go on. But it's going to take some time.
Yeah, I think we're just at the dawn of this, intersection of AI, bio, large data sets. I mean, we've talked about this a lot on the show. It's these, downstream analyses dependent on AI are so dependent on what you feed into the algorithm, right? Garbage in, garbage out, right? So you got to make sure that initial data set is good to make sure your downstream analyses or whatever also that the AI spits out are also good. And I think you're at a perfect place to be answering all these questions. You're at, of course, Children's Mercy. You moved there from the Broad at MIT, where, we talked about, you're the director of disease gene engineering. It's a big change in environment. different, moving from Boston to the middle of the country to Kansas City, but also kind of a representation of the transition of these genetic engineering approaches from academic laboratories to clinical practice. I mean, Children's Mercy is a hospital, right? I'm at Cedars-Sinai, of course, first and foremost, a hospital. And you're also in Kansas City, and when people think of Kansas City, they don't always think of high-powered research programs, right? They might think of, great barbecue, great football as the home of the infamous Kansas City Chiefs. But with Children's Mercy, we talked about the Stowers Institute before the show. It's also there in Kansas City Research Institute, UK Medical Center. There's a ton of incredible basic and translational work that's going on in this major hub of the Midwest. So talk about, where you are right now, Children's Mercy, talk about Kansas City and what makes this such a great place for doing the science that you're doing right now.
Yeah, absolutely. So Kansas City is wonderful. So I want to say nice things about Kansas City, but please don't interpret that as me saying negative things about anywhere else. Everywhere is great. But Kansas City is a really cool kind of community-centric city, right? So Children's Mercy, we're, a standalone pediatric hospital that has community support, and philanthropy is great, and just everything about the city is great. But also, I think it was important for me to step outside of the machine, right? So I did, when I was in Boston, I trained at Harvard, and then I went to the Broad as kind of a professional scientist, and talk about some of the best places you could possibly be and seeing the coolest things that you could possibly see. But figuring out how to translate those things in a way where the work that I'm doing on day to day, I see the impact sometime soon, can get kind of hard. And for somebody like me, I thrive more on the seeing things, actually happen rather than the perspective like, hey, 30 years from now, this is going to mean, change everything. So that was a big part of it. I wanted to kind of get out of that system, and again, the rare disease program that was kind of being discussed when I came here excited me because, again, rare disease is not this centralized problem, where we can just say this is going to be the place that does it and they're going to be the best ever. This has got to happen everywhere. So why not go somewhere and do it? And the other really cool thing, in a major aspect of our lab's success is being in a small hospital. So there's several sites of Children's Mercy, but our lab is in the main hospital, in downtown Kansas City, and it's one building. And so a lot of the work that we do is highly translational. It involves clinicians, neurologists. I see those neurologists in the hall all the time. We stop and we chat, right? It's just the communication is so seamless. Whereas, again, not to knock on anything, in Boston, especially towards my later years there, pre-COVID, like it was already to the point where meetings, down the street were already happening virtually because it just kind of everybody's too busy and it got kind of challenging to navigate the city. So let's just have this meeting virtually. And then all interactions kind of had to be scheduled and planned. And then having more of an organic kind of environment where we're all just clinical work, research work all happens kind of under the same roof. That just makes all these things synergize, and it makes the work more natural. So it just kind of organically happens. So I think it's a big component and something people should think about. Wanting to do highly translational research. Are you interacting with the clinicians? Are you seeing the patients? Like I get emails sometimes from parents of patients. Like it's that level of engagement that's super exciting, and it's extremely motivating for the work too, when you're talking to parents whose children you're trying to help.
Yeah, I mean, it's so everyone, I think, at this point appreciates how critical that those interactions, the little micro interactions, right? The stop and chats, and getting a critical mass of thinkers in one place to kind of rub against each other and let the sparks fly, right? But I hadn't thought about the intimacy of an institute where all the stakeholders, including the patients, have that interaction, because that's another kind of synergy that is not just, about it takes two or three to think, but it's about the inspiration underlying it. So I think it's a really important point. And I think, especially as Arun mentioned, as these, we translate these preclinical ideas from these big labs and major silos, right? As we spread them out across the world, I think it's really key to do it in places where we have that level of intimacy. So I'm really glad we got that insight from you and that perspective from you. It's really such a valuable view to share with our listeners. And it's not just business for you, Scott. We talked before the show about how you have a personal connection to rare diseases, and you're not the only one. A lot of the lab members do too. Tell us a bit about that, would you?
Sure. One of the big things about my lab, everybody that's come through the lab has been directly impacted by rare disease, whether it be through a child or a sibling. And that's not by design. That's just like it happened that way. And so even our son was born with a congenital heart defect. And so fortunately it was one that can be resolved by surgery, which is really kind of crazy. Like he was born in this hospital, had to have open heart surgery like in this hospital by a colleague of mine. So that kind of it's been really interesting to just see this, like it drives everybody in the lab on a personal level, which is super cool because I feel a little bit less like I've got to come in and be a cheerleader for like, hey guys, let's do this thing. It just impacts so many people. So it's been really interesting.
Before we let you off the hook here, we got some more sharing we're going to ask you to do on a more, maybe even more intimate level, more about you as a person, more than a scientist, perhaps, or maybe even not a scientist. As the first question begins, if you weren't a scientist, what do you think you'd be doing with yourself and your time?
So what would I want to be doing? This changes over time. I think kind of more in my adult life, something about being a chef seems really exciting to me. And maybe this is too many seasons of Top Chef, but just like the high intensity, the perfection. And this is in no way saying anything about my culinary abilities, but it's just a desire to be in the upper echelon of chefs. It just something about that is super exciting. And maybe it's exciting because I'm not a part of it, and you know maybe I'm romanticizing about it. But that's what I'm going to go with for now.
Oh man, I'm with you. I love to cook, and I really think I don't think I'd make a great chef, but I think I'd be a very happy chef if in an alternate life, alternate career. And I've thought about this deeply because I think the real satisfaction I get, at least from it, is that there's always an answer, right? As opposed to biology, where there's an objective truth, and we're all trying to get at it and maybe get a glancing blow and get really thrilled about it. With food, you get to eat it at the end of the day, even if it's disgusting. It's an answer.
Yeah, right. You chalk it up, whatever it was, you try it again tomorrow.
Yes, again, every time. My wife would tell you that I treat the kitchen like a lab and it drives her crazy. But I
Yeah.
would tell you that's just good chefing. Anyway, to the last question, a fill in the blank we have for you here. When I'm not conducting research, I am.
Yeah, I am at home with family. So we have a 7-year-old, and my wife and I have a 7-year-old and an almost 4-year-old. And I thought science was challenging, but it turns out parenting is just next level stuff that nobody kind of prepares you for. But it's also super cool because you have these I don't want to call them critics, but these individuals that live with you that evaluate your every move in ways in an unfiltered way that kind of keeps you grounded. So I enjoy spending time with them.
That's a funny way of putting it. I never thought that I was living with constant peer review with my
Yeah.
two sons, but that is exactly what it is.
Yes.
No one can really shrink you like your children. But I think that makes three of us, all with two kids. Arun, what do you have to say about the joys of parenthood now that you've got a nearing, what, 3-month-old?
Something like that. Who's counting? No, I like that. Peer review at home. It's not like we don't see enough peer review as it is, right?
Well, I think the review on this interview, call it a chat, intimate as it was, is going to be glowing. We're going to get through on this one, guys, because it was just a fun conversation. I hope our listeners will enjoy it. Get back to us with comments or criticisms, please. But Scott, really, thanks so much for joining us on the show today. This has been a little bit out of our wheelhouse, and that's how we like it, to learn a little bit and to share a lot. So thanks for joining us on the show today.
Yeah, thanks so much for having me and chatting about our work. It's always a pleasure to do so.
All right, you guys, that brings us to the end of our show for today. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCELL Podcast, or via email at info@stemcellpodcast.com with feedback or to suggest guests. Until the next time, thank you guys so much for listening. Looking forward to coming at you from the ISSCR in Hong Kong, T-minus 2 months.